Good afternoon, class. For this session, we're going to discuss cell-mediated immunity or cell-mediated immune responses. At the end of the session, the student should be able to differentiate humoral from cell-mediated immunity and innate cell-mediated from adaptive cell-mediated immunity. Identify the components of innate cell-mediated immune responses and give their functions. Discuss how T cells recognize antigen and how they are activated. And finally, discuss the various subsets of T lymphocytes and their functions. Immune responses are traditionally classified as humoral or cell-mediated. By humoral, we mean there is no actual cell-to-cell contact between cells of the immune system and their targets. On the other hand, when you have cell-mediated immune responses, there is an actual cell-to-cell contact between the cell of the immune system and its target. In the case of humoral immune responses, molecules mediate the interaction. Another way of classifying immune responses would be whether they are innate, which are very early responses, the moment you encounter a pathogen, for example, and set in immediately till about 96 hours following initiation of events. On the other hand, adaptive immune responses take some time and play a more prominent role beyond 96 hours. What we're going to discuss in this lecture are cell-mediated innate immune responses and cell-mediated adaptive or specific immune responses. One of the important molecules that is produced during an immune response would be, for example, a cytokine. Cytokines are molecules derived from single cells and can be classified in various ways. This is one classification. They can be classified as interferons. The interferons actually are antiviral. They regulate innate immunity and activate antiproliferative effects, and they have antiviral properties preventing viral replication. Interleukins are cytokines that mediate the growth and differentiation of leukocytes. Many are pro-inflammatory. Chemokines, on the other hand, are taking control of chemotaxis. They attract inflammatory cells into areas of infection, for example. Colony-stimulating factors like G-CSF and GM-CSF stimulate hematopoietic progenitor cell proliferation and differentiation. And tumor necrosis factor are pro-inflammatory cytokines that can activate certain types of killer cells like the cytotoxic T lymphocyte. Now, cytokines are produced during the innate immune response from various types of cells. These cells include cells of the immune system, as well as other types of cells not traditionally classified as belonging to the immune system. Now, one of the reasons why cytokines are able to be produced from these cells is because immune cells and other types of cells may have what we call pattern recognition receptors. The pattern recognition receptors may be found outside the cell or inside the cell. They recognize pathogen-associated molecular patterns. These are conserved structures that are found across a variety of pathogenic organisms. They can also recognize damage-associated molecular patterns, just in case host cell tissues and cells are damaged. The end result is initiation of transcription factors that result in the production of cytokines. Here on this diagram, you see TLRs or toll-like receptors that recognize certain components of the bacterial cell wall, for example. There are also cytokines that are found inside the cytoplasm and attached to the endosomes. Toll-like receptors can also be endosomal, in which case they are able to recognize microbial DNA and RNA that are found in the endosomes. Those that are found on the in the cytoplasm include nucleotide oligomerization-like domain receptors, or NLRs, or NOD-like receptors. You also have RLRs, or RIG-like receptors involved in interferon production. And you have a variety of other cytosolic sensors. The most well-described pattern recognition receptors are the toll-like receptors. And there are various toll-like receptors found on the plasma membrane or on the endosomes. And they recognize various types of components, like TLR4 can recognize gram-negative bacteria, especially endotoxin. TLR5 can recognize bacterial flagellin. TLR2 and 6 can recognize components of gram-positive bacteria and fungi. Those that are found on the endosome can recognize viral double-stranded RNA, single-stranded RNA, viral proteins, as well as bacterial or viral DNA. An important cytokine which is produced early in viral infection is interferon. Now, virus-infected cells, for example, certain lymphocytes, fibroblasts, when they get infected, they're able to produce interferon. There are three types of interferon, but what we're talking about here are the so-called type 1 antiviral interferons, exemplified by interferon alpha and beta. Interferon lambda, which are type 3 interferons, are also antiviral. Now, interferon acts on cells which are not yet infected by the virus, so that even if that cell gets infected by the virus is unable to replicate inside the cell because interferon degrades viral RNA and stops viral protein synthesis. So early in the course of a viral infection, production of interferon helps control viral proliferation. Now, NOD-like receptors, for example, are able to form what we call inflammasomes. These are large molecular complexes that are formed with pro-caspases and other proteins. An example is the NLRP3 inflammasome. When it is formed, its job is to cleave the proforms or inactive forms of interleukin-1 beta and interleukin-18. As a result of the inflammasome activation, you produce interleukin-1 beta and interleukin-18, which are pro-inflammatory cytokines, meaning to say they enhance inflammation. Cytokines are very important, and they represent humoral immune responses, but these cytokine production have many effects. Cytokines have pleiotropic effects. They can affect various organs. For example, they are the principal reason why you may have fever when they affect the hypothalamus. They can affect, for example, the adrenal glands, causing increased production of steroids. They can enhance migration of inflammatory cells and leukocytes from the blood vessels to site of infections. They can cause the liver to secrete acute phase reactants CRP, which can rise during an infectious process. So these are examples of humoral innate immune responses that occur very early. On the other hand, just to differentiate, we have what we call innate cell-mediated immune responses. The primary example, which you may perhaps know already, is phagocytosis. We have certain cells which can swallow up organisms or pathogens. These include neutrophils, macrophages, and special types of cells we call dendritic cells. These phagocytic cells are able to take up foreign material, including pathogenic organisms. Phagocytosis is often followed by intracellular killing. However, this is not always successful as there are certain organisms which can evade intracellular killing, and some of them are heavily encapsulated, making them difficult to phagocytize. There are various steps in phagocytosis, but a key step would be recognition as well of pattern recognition receptors recognizing PAMPs on the organisms, on, for example, bacterial cells. So this causes adhesion to the target bacterial cell, followed by engulfment into a phagocytic vacuole with the formation of a phagosome. Now, we have certain organelles called lysosomes which contain various destructive enzymes, and they can fuse with the phagosomes. So there is phagolysosomal fusion. This is followed by release of the various enzymes that result in killing of this and destruction of the bacterial cells that are taken up inside phagosomes, followed by release of residual debris. The enzymes involved include lysozyme, DNase, RNase, proteases, peroxidases, and other products such as reactive oxygen intermediates, including superoxide, hydrogen peroxide, and singlet oxygen. So, phagocytes include the neutrophils. But in addition, neutrophils that are dying can release web-like structures called nets or neutrophil extracellular traps, which are composed of cellular DNA that are coated with proteins, which are also derived from the granules found inside the neutrophils. So the nets can additionally trap or kill bacteria. So here you see an example of an innate cell-mediated immune response because a phagocyte has to attach to a target in order for phagocytosis to take place. That attachment is aided by pattern recognition receptors. Another cell that participates in innate cell-mediated immunity or cellular immunity would be what we call natural killer cells. These are a type of innate lymphoid cells. They are lymphocytes, but they are not B lymphocytes, neither are they T lymphocytes. They are slightly bigger than B or T cells, and their job is to kill cells that look foreign to the immune system. And these cells that look foreign are usually cells that are infected by viruses. And they also include tumor cells or malignant cells that display different types of antigen. Now, the natural killer cell contains destructive enzymes like perforins or granzymes. So perforins and granzymes are released from natural killer cell when it makes contact with the target cell. In the illustration, the small cell with the arrow pointing to it is a natural killer cell, and you see the big cell, which is the target. Down below on the right side, you see the small cell attaching to a target cell, about to release its contents, and these contents are perforins and granzymes that attack target cells, leading to cell death. Now, to a certain extent, natural killer cells recognize our own cells and they don't attack these cells that we normally have. And the reason is because they have receptors that recognize self-antigens. These self-antigens are found on all cells that are nucleated. They are MHC molecules or major histocompatibility complex molecules. The one that you find on all cells that are nucleated are classified as MHC class 1 cells. When a natural killer cell is able to recognize it, then the natural killer cell does not attack that cell. On the other hand, for example, when you have a viral infection, viruses can downgrade the expression of MHC class 1 molecules. So when that is absent, what is recognized instead is different from the MHC class 1, and it results in the activation of a natural killer cell. The natural killer cell is able to produce inflammatory cytokines like interferon gamma and tumor necrosis factor alpha, in addition to the enzymes that it's able to release, like perforins and granzymes, resulting in destruction of the target cell. Later on, following the production of specific antibodies, natural killer cells may also have an additional function. The natural killer cell attacks cells indiscriminately, so they are broadly specific. So whether a virus is different from another and infects different cells, the natural killer cell attacks it in the same way. On the other hand, when you already have an antibody later in the course of infection, the antibody can attach to specific antigens presented on a target cell, and the natural killer cell can attach to the antibody molecule, resulting in an antibody-dependent cell killing or antibody-dependent cellular cytotoxicity. So we have discussed innate cell-mediated immune responses, and the most important examples that I gave are phagocytosis and natural killer cell activity. They are innate immune responses. They are ready at hand the first time you encounter an organism. So they are very important. But the reason for their importance is without innate immune responses, you would be quickly overwhelmed by an infection. However, as I mentioned, innate immune responses are broadly specific and they are non-amplifiable, meaning to say the second time you encounter an organism, you hardly develop any memory of the organism. We call that immunologic memory. And so the level of response the second time around is similar to the first time around. So these are limitations of innate immune responses, but without innate immune responses, you might perhaps die of severe infection. On the other hand, when you have adaptive immune responses, these are specific immune responses. Later on in the next lecture, you're going to discuss antibodies, which is a specific type of humoral immune response. But for this lecture, the focus is on adaptive cell-mediated immune responses, which are mediated by T lymphocytes or T cells. The B lymphocytes, when they leave the bone marrow, are already B cells. On the other hand, when the T cells leave the bone marrow, they are not yet T cells. We call them pre-T, Pre-T T or pre-T cells. They move on to the thymus, which is an organ present in the anterior portion of the superior mediastinum, where they develop. And as they develop, they undergo two types of selection. The T cells initially develop receptors on their surface. They also develop various molecules on their surface. We call them CD molecules, cluster of differentiation molecules. The reason why they're called cluster of differentiation molecules is that using, for example, a technique called flow cytometry, you can identify certain cells as belonging to certain types or clusters or subsets. So these are named by numbers. So you have CD3, CD4, CD8, and so on. Now, in the thymus, a T cell develops a lot of these CD4 molecules and a lot of T cell receptors. Now, if the developing T cell recognizes your own self-antigens or MHCs, MHC molecules, they successfully pass thymic development. But however, there is a second type of selection that goes on. The T cells must not react to your own self-antigens, because self-antigens from various cells and tissues can get presented in the thymus to the developing T cells. And if the T cells are able to attack these targets, they are deleted. So only a few of T lymphocytes that get into the thymus successfully get out alive. Now, when they leave the thymus, they can either have CD4 or CD8. So we call them CD4 positive T cells or CD8 positive T cells. The T cell that is CD4 positive but CD8 negative is called the T helper cell, whereas the CD4 negative, CD8 positive T cell is called the cytotoxic T cell or cytolytic T cell. Now, in the peripheral circulation, the level of CD4 T cell is higher than CD8, and the ratio is about 1.5:1 or 2:1. When you look at the cross-section of the T cell surface, this is what you're going to find. The black colored chains here in the middle are what you call the T cell receptor, consisting of two types of chains, an alpha chain and a beta chain. There are certain T cells that contain gamma and delta receptors, but majority of T cells have alpha and beta T cell receptors. The T cell receptor is surrounded by what is called CD3, which happens to be an important molecule. It's found on all T lymphocytes and acts as the signal transducer. When the T cell receptor is able to recognize the corresponding antigen, what happens is that CD3 gets activated as well and sends a signal to the interior of the cell. It initiates the chain of intracell signaling. Now, some cells have CD4, as I mentioned, some have CD8. And in addition, there are other CD molecules like CD28. So the reason why the T cell or T lymphocyte is able to recognize antigen is because of the T cell receptor. However, unlike the receptor of the B lymphocyte, which is surface IgM, the T cell receptor is unable to recognize free or soluble antigen. In order for it to recognize antigen, the antigen must first be processed by what we call antigen-presenting cells. Here are the professional antigen-presenting cells. The dendritic cell, which may be found initially in the skin, for example, when they pick up antigen, because they may initially be phagocytic, they may subsequently migrate to secondary lymphoid organs where they present this antigen to T lymphocytes. Other cells include macrophages, which we have already encountered a while ago, and the B lymphocyte, because the B cell, although it is not phagocytic, is able to endocytose or take up foreign antigen, process it, and present it as well to the T cell. The dendritic cell that is most active in antigen presentation is what we call the conventional dendritic cell. There are certain types of dendritic cells called plasmacytoid dendritic cells that are, on the other hand, involved in interferon production. The dendritic cell is very important, and the reason for its importance is because you need the dendritic cell for initial activation of the T lymphocyte. Subsequently, however, after they have been activated, macrophages and B lymphocytes can also present antigen as well. Now, the antigen-presenting cells need to process the antigen first and present it on MHC molecules. So MHC molecules are very important. I already mentioned a while ago MHC, the major histocompatibility complex type 1 molecules. These include what we call HLA-A, B, and C. And these MHC class 1 molecules are found on all nucleated cells, including the professional antigen-presenting cells. How does an antigen-presenting cell process MHC, process peptide, and then brings it to MHC class 1? So there are certain peptides that get inside the cell. For example, when this cell is infected by a virus, viral replication happens inside, and so you form viral proteins. These viral proteins can be broken up into small pieces and processed by what we call a proteasome. They are then brought to a tap transporter, and through that way, they get into the endoplasmic reticulum where your MHC1 complexes are formed. So you form MHC1 peptide complexes in the endoplasmic reticulum, and these are then brought out to the cytoplasm and then to the surface of the cell. So the MHC1 plus peptide can be displayed eventually on the cell surface. And these peptides are coming from the intracellular pathogen like a virus. This can then be presented to a T lymphocyte. Now, what about for organisms that are found outside the cell? Well, antigen-presenting cells can take them up as well, for example, by phagocytosis. So proteins may get inside such a cell. So these peptides are found in phagosomes. Now, the MHC2 molecule is quite different from MHC class 1. The MHC class 2 molecules, which include HLA-DT, DQ, and DR, are found only on immune-competent cells. So the professional antigen-presenting cells, for example, have MHC class 2 as well. The MHC class 2 is complex to an invariant chain in an inactive form in the endoplasmic reticulum. These are then brought out into endosomes, and the invariant chain is subsequently cleaved and replaced by the peptides that are found in phagosomes following endolysosomal fusion. What happens after this is that the peptide links up to the MHC class II and is the one displayed on the cell surface. So here the difference is that your protein that is broken up into peptides comes from outside the cell, whereas in MHC class I processing, what happens is you usually have an intracellular organism which is processed. The dendritic cell is the important cell that initiates T cell activation. So it's a very important cell, and it straddles the divide between innate immunity and adaptive immunity. It's also capable of producing cytokines, and depending on the infecting type of organism, the cytokines that can be produced may be different, and the cytokine milieu and the type of organism can actually influence the T cell to divide or proliferate and form different subsets. So they influence not only T cell activation, initial T cell activation, by initiating it, they also provide an important milieu for T cell development into different subsets. Now, the way antigen is presented to the T cell receptor on top of an MHC molecule is like this. Here, you see an MHC molecule in yellow containing on top of it the peptide antigen. It's recognized by the T cell receptor. But notice, however, that recognition involves CD4 molecule as well on the T cell. In this case, because it has CD4 molecule, this is a T helper cell. And the requirement is that the antigen, before it can be recognized by a CD4 T cell, must be presented complex to an MHC class II. So that is an MHC restricted presentation or recognition. On the other hand, if you're dealing with a CD8 T cell or cytotoxic T cell, the peptide must be complex to MHC class I. So there is a difference in MHC restriction. So not only do you need T cell peptide MHC recognition or activation, you also need the co-stimulatory molecules. For example, CD80 or 86 complex to CD28. And then you have also the cytokine milieu. So here is a comparison of the interactions that are present when a T cell gets activated. On the left side, you have a CD4 positive or helper T cell. On the right side, you have a CD8 positive or cytotoxic T cell. Colored yellow here would be your antigen presenting cell, and then on the left side of each diagram, you have your T cell, CD4 or CD8. So important for initial activation would be the interaction of your T cell receptor with the peptide MHC complex. But notice that you also have interaction of other co-stimulatory molecules. For example, CD80, 86 on the antigen presenting cell and CD28 on the T cell, ICAM1 on the antigen presenting cell, and LFA1 on the CD4 T cell, LFA3 on the antigen-presenting cell and CD2 on the CD4 T cell. So this makes the interaction between the peptide MHC complex and the T cell reactive more secure, and you need the co-stimulatory signal for initial T cell activation. If you don't have the co-stimulatory signal, the T cell may not be activated and goes into a state which we call anergy, Anergy. On the right side, you have your interaction between a CD8 T cell and an antigen-presenting cell. This time, you have an MHC class I peptide complex being recognized by the T cell receptor. This is accompanied by other co-stimulatory signals like LFA3, CD2, CD8086, CD28, similar to what we discussed previously, ICAM1, LFA1. Now, we said that for initial T cell activation, the important cell is the dendritic cell. It seems that when you have activated the T cell initially, they can recognize other peptide MHC complexes subsequently without needing co-stimulatory signals. So the co-stimulatory signals are important for initial T cell activation. Now, recently, one way by which the cytolytic or CD8 T cell can get activated has been recognized. In this diagram, you see naive or inactivated CD8 T cell on top. So you need a signal to activate it. And one signal is provided by the interaction of T cell receptor with the MHC class I peptide complex, since it's a CD8 T cell. Another signal is provided by the interaction between CD28 and CD8086. However, it seems that the presenting cell here is shown in yellow, the dendritic cell, has to be helped by the T helper cell or a CD4 T cell belonging to a particular subset, Th1. The help provided by the Th1 makes the dendritic cell able to activate the cytolytic T cell. What happens when the cytolytic T cell is activated and when the helper T cell is also activated? It results in production of cytokines of various types, and it results in T cell proliferation. T cell proliferation results in the development of effector T cells. But not only do you have effector T cells, you also develop memory T cells, of which you have various types. The stem cell memory T cells, which are found in the primary lymphoid organs like the bone marrow, also the central memory T cells, the effector memory T cells, and the terminally differentiated cells. The memory T cells serve an important role because the effector T cells last only for a short period of time. The memory T cells last for longer periods of time, maybe months or years, in contrast to effector T cells. Now, as a result of T cell activation and T cell help provided to B cells likewise can develop immunologic memory. So you have central memory B cells as well, which are long-lived. You also have long-lived plasma cells because eventually when a B cell is activated, it develops into an antibody-producing plasma cell. But some of these majority of these plasma cells are short-lived They produce antibody only for a short period of time and then they die. But the long-lived plasma cells go back to the bone marrow and may be responsible for longer-term production of antibodies. But you also have central memory B cells. The T cells, as I mentioned, you can have memory T cells. You have central memory T cells, effector memory T cells that quickly develop into effector T cells, and you have resident memory T cells that go to the various tissues, and when they encounter the antigen that they recognize initially, they eventually develop into effector cells. So immunologic memory is very important. It is what is responsible for the more rapid response that you see the second time you encounter an organism. They are specific to the organism that initially stimulated them, and it's for this reason that vaccines can have a booster response because you develop immunologic memory. Now, we said that as a result of T cell activation, you have the formation of effector T cells. So, what are the effects of these effector T cells? They produce a variety of cytokines with different functions. And depending on the type of cytokine that they produce and the type of function that they exert, helper T cells, for example, or CD4 positive T cells, can be divided into several subsets. For example, you have peripheral regulatory T cells. They exert a suppressive function. Their effector cytokines are interleukin-10 and transforming growth factor beta. They are capable of suppressing immune responses when you no longer need these responses. Let's say you got over the infection, you need to suppress active immune responses. That's the job of the T regulatory cell, which is a subtype of a CD4 T cell. The Th17 cell, which produces interleukin-17 cytokine, is important for extracellular pathogenic defense, like extracellular bacteria or fungi, because they these interleukins recruit phagocytes like neutrophils into areas of infection. So that's the job of the Th17 cell and the cytokine interleukin-17 that they produce. The Th2 cell produces interleukin-4, interleukin-5, and interleukin-13. They are important activators of eosinophils. They are important for helminthic infections, and they play a role in the development of allergy, since interleukin 4 or 5 can actually switch production of B cells from certain types of immunoglobulins to IgE, for example, which is involved in allergy. Now, there are other T cell types like Th1, second to the right, which is important by producing interferon gamma. Interferon gamma combats intracellular pathogen because it's responsible for activating macrophages and enhancing intracellular killing. So that's the job of your Th1 cell. The TFH cell, or follicular helper T cell, is another type of CD4 T cell. It's usually found in the germinal centers of the secondary lymphoid organs, and they regulate affinity maturation in the germinal center B cells. So essentially, they have a job similar to Th2 cells, which are found in the periphery. The follicular helper cells aid humoral immunity by making the B cells produce better antibodies, and as a result of their cytokine production, they can also stimulate switching of antibody production. There are other newly discovered T helper subsets like Th9 and Th22. Going back to the Th1 cell, and we mentioned interferon gamma in connection with it, this is one particular type of infection for which it is important. Mycobacterium tuberculosis, or TB. Here you see a macrophage being infected by TB bacilli. So you see mycobacteria inside. Now, mycobacteria can evade intracellular killing. However, some of them get killed and processed, and so your macrophage, which is also antigen presenting, can present mycobacterial antigens to a Th1 cell or T helper subset 1 cell. Th1 produces interferon gamma, and the interferon gamma enhances the killing of mycobacteria inside. So interferon gamma is important in this way. And you see the job of a Th1 cell. The Th2 cell and the TFH cell or follicular helper cell are important in this way for the production of memory B cells, immunoglobulin isotype switching, production of antibodies with better affinity or binding and avidity to target. So here you have a B cell presenting antigen to a T cell. The T cell is able to produce cytokines that tell the B cell to proliferate and divide and produce memory B cells. And once they develop as well into plasma cells, they are able to switch production from IgM to, let's say, IgA or IgE and IgG. So you call that Ig immunoglobulin isotype switching. And as I mentioned, the antibody that you produce when you have T cell help would result in better binding to antigen. So this is a function of your Th2 and the TFH cell, which are found in, the latter is found in the germinal centers of the secondary lymphoid organs. Now, what about the cytotoxic T cell? The cytotoxic T cell or killer T cell, recall that it's different from the natural killer cell that we discussed in the early part of the lecture. So these are T cells. The cytolytic or CD8 T cells have one specific function, that is to kill cells that are infected by viruses. So they do this through apoptosis or programmed cell death and by perforin granzyme mediated destruction. So we encountered perforins and granzymes when we discussed natural killer cells, which is part of innate immunity. But these same enzymes are involved and are produced as well by the cytotoxic T cell and are released to kill an infected cell. So why do you need to kill an infected cell or a cell infected by the virus? It's because when a cell is inside the virus, when a virus is inside the cell, there is no way to get rid of the virus without killing the cell. So you see that cytotoxicity generates some sort of cytopathology and may be responsible for some of the unwanted effects that you see in terms of cell death or necrosis that you find in infected tissues or organs. Apoptosis, on the other hand, is defined as programmed cell death. For example, when you have interaction between Fas ligand and Fas, which are found on the cytotoxic T cell surface as well as the target surface, the interaction sends a signal to the interior of the target, causing the release of caspases and destruction of DNA. And when DNA is destroyed, the target cell dies. So this is another way of T cell killing. So you can understand why MHC class 1 molecules are found practically on all our cells that are nucleated because that is any cell can become infected by a virus depending on the type of virus, and therefore there is the potential for any cell to be able to become a target of a cytotoxic T cell. So we have discussed cell-mediated immunity, both innate and adaptive. To summarize, innate cell-mediated immunity involves phagocytes. So phagocytosis plus natural killer cell activity. Natural killer cell activity results in destruction or killing of cells that are infected by viruses. Now, the same thing can be accomplished by cytotoxic or CD8 T cells, but the difference is that cytotoxic T cells are narrowly specific to certain targets or antigens, whereas natural killer cells are broadly specific and come early on during the innate immune response, whereas the cytotoxic T cells are part of the subsequent adaptive immune response. The adaptive immune response, which is cell-mediated, also involves a variety of subsets of what we call T helper cells. And the T helper cells have many functions depending on the type of cytokine that they produce. Eventually, what decides whether what type of T helper cell or CD4 T cell will predominate in an individual during certain phases of life can be influenced by the type of organism that you encounter and the cytokine milieu that is provided in the environment, with the dendritic cell providing a very important role for initial activation. Thank you very much.